Oops. Hi, Dustin.
Hi.
We have a microphone today. Yay.
Yeah.
How are you doing?
I'm doing well. How have you been?
I'm good. I think if I could surf and hike, I'd actually be okay.
Are they cracking down on it a lot over you're in La Jolla, right?
Yeah. Oh, I've been in the water for a couple weeks. Probably 3 now,
Oh, wow.
and I managed to find a trail that crossed a road east of Sunrise Highway on Saturday to get out for a little bit.
Yeah.
But yeah, hiking doesn't seem uh it's not, I guess it's not encouraged, and people are too concentrated on the trails that are accessible.
Yeah. It was raining a lot last week. It
Oh geez,
made it
yeah.
like terrible.
Yeah. We went to one trailhead and I'm like, "Oh, there's too many people." And then we went to another trail and there was even worse. And then we found this spot where the trail crossed the road and it seemed kind of remote, and it was really remote. It was a dirt sort of sketchy muddy road. We got out, we went walking, hear a waterfall. We started to try to go find, walk up to it, but it was really dense. And I completely submerged my foot and boot in the
Oh,
water. And I looked at my watch and it was 2 minutes and 45 seconds since we had left the car.
terrible. Um,
I had good socks on though, so it was fine. We stayed out for another couple hours. But it's
oh,
like, that's gotta be a record of some sort.
I can't stand like wet socks.
These were Smartwool. It was squishy for a little while, but I mean, I was walking around in them for 3 hours and I didn't have any blisters or anything.
Okay, that's good.
Yeah. I wonder if we're gonna get anybody else today.
I would think so since we have a test coming up, but
I know, yeah. So the instructions were clear, right? 'Cause
yeah, I
I have to
felt like
send
it.
s okay, I have to send so many emails and I forget, like, I think I had to resend the lab one yesterday for my Chem 152L people, 'cause I think I either didn't send them the new, access information, or I sent the wrong one. They were all having trouble getting in. I'm like, it was 5 minutes after, I'm like, "Nobody's here?" So it's an easy lab, but and then I looked at my emails like, "Oh."
I don't think it didn't ask me to put in a password, so
It didn't? Well, it's weird because I got asked to put in a password.
maybe since like the link you sent us is like private, so we don't need to put one in. I don't know.
Yes, so that could the one I had to put in a password for was for a yoga class, but I clicked on the link they sent and I had read something that, yeah, it creates a link that if you click on that exact one, no password is required.
Okay, so that's probably the one you sent because that's all I did was click on the link in the email and it opened right up.
Cool. So, where are you on the homework?
Um, I've gotten through most of it.
Okay. It's a slog, but I feel like it's good for it's just good for encouraging time with the material in some level of depth, you know?
Yes.
Because
And
this I find this book hard to read, just to read.
yeah, there are definitely parts of it where it's just I find myself just, going through the motions, like reading it but not really taking it in because it's just really dense and
It's
boring.
very dense. Yeah. And I get it, like with in translation when they have all the elongation factors, and especially with the eukaryotic stuff when there's 18 proteins involved.
Yeah, that's
And if they're going to write about the process, they should put where they know each of the factors are, but it's almost like they should have just the generic paragraph that doesn't include the details of which factors where, and
Yeah.
then the one where it's in more depth, but that's a lot of work. Okay, let me see. I'm gonna share a screen. This has been like a surprisingly non-interactive class. Usually my classes are very talkative. Okay, do you see my PowerPoint?
Yes, now I do.
Okay, yeah. Yeah, well, and the other thing is that students have contacted me afterward to see the Zoom thing. It's like, well, if no one's here, there's no questions for me to help. So, the main objective for today is to answer questions about Chapter 12 homework. Most of it is, I think, um, reading in detail with the possible exception of, I thought the wobble stuff was a little
Yeah.
bit confusing.
Some of that was like, I think I had a couple questions on that, and that was pretty much the only stuff that the wording of some of those questions just kind of threw me off.
Yes. Well, and yeah, I agree because I looked at it, I was like, what? And then um and then I have the instructor manual and I read the answer to one of them and I think that's wrong. Like there was just one sentence in it that was wrong. Um but yeah, it's a little bit, and it is challenging because there's so many reversals and templates and five prime to three prime, three prime to five prime kind
Right.
of stuff. You can get turned around. All right. Well, usually got questions too. Maybe he'll come in later. All right. Well, so for the exam, we've already talked about this. It'll be essay style.
Mhm.
I want you can type answers, but not no if there's going to be a figure, it should be drawn by hand,
Okay.
drawn from and I'll try to draw it generally from questions that we have focused on. It's open book. Um, I used to do this when I taught this material back in 1998. um, I like to do this section like where I write something down like the Shine-Dalgarno sequence, and then the question is it protein? Is it DNA? Is it RNA? What kind of RNA? Is it tRNA? Is it messenger RNA? And then define what its function is. And um, I was surprised at the time to realize how many people didn't realize, oh, we're talking about protein. I should probably also include like small molecule in there. Or, oh, we're talking about RNA. Oh, wait, this is DNA, that kind of thing. That forces you to think in a little bit more detail about the process.
Process, yeah.
Yeah. And then questions from the homework, but because it's open book, there'll be a little bit more comparison work. And for example, one question I'm thinking of is, and things in this kind of style is describe translation initiation in prokaryotes, and then describe how it's different in eukaryotes. So
Okay.
we spent a lot of time going over the prokaryotic processes and really have just drawn comparisons to eukaryotes. And I think that's fine because the eukaryotic ones are so complicated. But if we can identify the major points of difference, then that's useful.
Yeah.
And I'm pretty comfortable with that format for the exam. And
How many questions is it gonna be?
well, I was going to look and see what I had for point values. Let me see. I'm thinking like 7 or 8. I think let me look at I also have to update the syllabus because we don't have in-class participation. So the case studies aren't going to work so well.
Yeah.
Although I think it would be so neat to go over some stuff with viruses right now. And we have that coming up in the schedule. Okay, so the exams are 75 points each. So yeah, I'm thinking like 7 or 8. Well, that's kind of they wouldn't all be long questions. probably a maximum of 8 questions. Some of them shorter than others, some of them a bit more, maybe a bit longer, like the one I just mentioned about initiation would be a longer question.
Yeah.
Okay, well, why don't you ask me questions about the homework,
Um, I
the
think I had
private class?
a
Oh, I should get the book. Do you know which one you were thinking of?
the first one that comes to mind is like number 8, where it says like it is possible for the codons for a single amino acid to have the first 2 bases in common and to differ in the third base. Why is this experimental observation consistent with the concept of wobble?
Oh, yeah. And so a lot of times these questions they're kind of open-ended, but this really has to do with binding. So it's possible for codons. So when the tRNA comes in and binds to the messenger RNA, the anticodon loop has a more specific association with the first 2 nucleotides in the codon. In the third position, there is flexibility with what it binds, right? Like inosine can bind A, U, or C,
See, yeah.
right? And so there has to be space to allow the tRNA to move around to make that to make that base pair with the third nucleotide. And that flexibility is what the term wobble comes from. So yeah, that's basically what it's about. The having
Okay.
flexibility.
So just
Go ahead.
no, I don't no. I was just kind of confused about like what it exactly is trying to ask, because I understand like that the third base pair has like the wobble ability, and that also helps because like the third codon typically is redundant in like most of the.
There's multiple ones that code for the same amino acid, you mean?
Yes.
Yeah. Um, yeah. And so you can think of wobble in terms of the ability to base pair with different nucleotides, or you can think of it mechanically, right? Like there's jockeying inside the ribosome.
Okay.
That's what this question was asking for.
Okay.
I'll have flexibility in when I read it from different people, because I think it can be interpreted different ways.
Okay.
Yeah.
Um, find the other ones.
No problem. It's been kind of fun for me to get reacquainted with this material, and it's somewhat satisfying to see how much I remember from 98.
For number 23, do you want us to be describe like both the prokaryotic and eukaryotic initiation complex, or
No, I think prokaryotic's fine. It didn't say, did it?
no? It just asks about the initiation complex.
Yeah, just I think in the book it was probably in the section that was prokaryotic. So prokaryotic's fine.
Okay. Uh,
I thought it was interesting that in the prokaryotic system, the messenger RNA binds first, and in the eukaryotic system, the tRNA arrives before the messenger. Isn't that weird?
yeah,
I guess if it's always the start tRNA, it doesn't matter.
I guess, yeah. And well, also probably because like prokaryotic, it can take place at the same time as transcription.
Mm-hmm.
So then like they can just bind right away and then not have to have like the amino acids present. Does that make sense?
Yeah.
Because
Well,
that way,
there's like the messenger's already like tethered, right? It's sort of lassoed by other ribosomes, but in a eukaryotic system, the messenger RNA is, it's on arrival. It's going through this process and floating around. So
yeah,
there might be tRNAs around before the messenger gets there, maybe. I don't know.
yeah, like the cell could be like ready with the tRNA already in place instead of waiting for the messenger RNA that needs to be processed.
Yeah. Maybe it's different rate limiting step.
Um,
I did like question number 42. How does it improve the efficiency of protein synthesis to have several binding sites of tRNA close to each other on the ribosome?
well, it can proceed like faster. It doesn't have to wait for.
Yeah, well, if we can definitely, it can proceed faster. Um, and it relates to what we learned about catalysis. Like one of the types of catalysis and enzyme catalysis is proximity and orientation.
Yeah, so it
There's
brings them close together in like the right position for that
exactly,
hydrolysis of the peptides together.
exactly, yeah. And it sort of gets them staged.
Um,
I never heard about a different start codon till this.
yeah, I didn't know that either. But then it was, I think it was like some random strain of algae or something. I can't remember.
It was in the MHC complex, and I think so it's part of the immune response, and I think it was one mammalian cell type, but I'm not totally sure.
Uh, for the diagram for like the first question, the flow chart for like protein synthesis, like how detailed? Um,
Not very. I think what I wrote for I do these myself too. Um, what I wrote is amino acid activation, then attachment to the tRNA, and then chain initiation, chain elongation, and termination.
okay, so we don't need to like start at like the transcription level, just
Mm-mm.
okay.
Yeah.
Um,
Oh, and then in terms of administration, the exam will be, I'll post it just like the homeworks get posted. So it'll be an assignment.
okay. Will it be like on the Dropbox or open it through like Canvas?
through Canvas.
Okay.
And I've had them in Canvas, right?
Yeah, to submit them, but the actual like prompts have been in.
Oh, no, yeah, I'll attach it to the Canvas
Okay.
assignment. I discovered the unfortunate difference. I gave midterms last week in my general chemistry class, and I created a module and I structured the written part like a quiz where the first question was where they take a pledge that they're not going to use the internet or talk to someone during the test, and then the second question they upload the file and then they work on it, and then they download the file, they work on it, and then they upload it. And when it's a question in a quiz, Canvas handles it completely differently than when it's an assignment being submitted that way. So like the speed grader doesn't work. I have to download it, open it in Acrobat, mark it up there, and then upload it to Canvas. Whereas with you guys, I just click on it and there's opportunity for me to make comments and to mark it up, the homework. I was so mad because there's 48 students in that class. And it made the grade
Terrible.
significantly longer. I don't know why it wouldn't be the same as with the assignment.
I don't know. I don't really have any other questions.
Okay, that's fine. If you think of anything later, let me know. Send me an email. I can resume if needed. It doesn't seem like it will be though, but I'm
No,
happy
most
to.
of the stuff after like the wobble stuff was pretty, I don't know. I feel like you go over it a lot in every biology class, and then it was, I don't know, you just have to read and
Yeah,
self-explanatory.
you have to spend time with it
Yeah.
and let it soak in. I'm grateful that there's a lot of repetition in the text, and I'm actually grateful for it because I haven't had biology since my freshman year in high school, even though I'm a biochemist.
Oh wow.
I managed to find a biochemistry degree in undergrad that did not require biology. It was a disservice to me as a scientist, actually, but that's what I did.
That's the thing I would like because I find the biology part really rather boring. A lot of memorization. I like the chemical part.
Me too. I don't find the biology part boring. I find it overwhelming. Like when you get to cell signaling and when
Oh,
there's 18 different proteins involved in translation, that kind of stuff. Because I feel like with my chemically oriented mind, I want to understand it in some kind of physical sense, but
yeah.
it's too much to take in all at once.
Yeah, trying to picture where all the 18 different proteins go. It's just, oh man.
Yeah. Okay. Well, so, oh, and the other thing for the exam is that we'll have, I'm trying to decide if there should be a time limit or not. It's probably better for you guys if there is, because I don't want you to spend, if I make it totally open-ended, you're going to feel obligated to spend too much time on it. So I'm thinking probably 2 hours inside a 24-hour window.
That sounds fair. Yeah.
And I'm flexible. If you guys feel like you need more time, I'll just make more time. But I think I want to start by setting expectations so that you don't
Spend
exhaust
forever.
yourself trying to do too much.
Okay. Yeah, the only thing I was worried about is every time we have to like do upload drawings,
It,
sometimes it doesn't work very well.
yes, that's exactly what I had happen with the general chemistry class. They were freaking out over uploading their images because well, they were probably taking it on their laptops, but then they needed to use their phones to do the imaging, and it was very poor guys. They were stressed out over it. They did well, but but it was trial by fire for everyone.
Okay, we'll learn from it and get better. So,
Yeah, yeah. Okay. All right. Well, then I'll sign off. And let me know if you need anything else.
yeah, I'll make sure to email you if I have any other questions.
Okay.
All
Stay
right,
healthy.
I'll try. You too. Thank you.
Thank you. Bye.
Bye.
See.